Good evening, everyone. So I'm Manchu. So today I'm going to take first tutorial for the course, um, Basics of Biology course. So the first question for this session, that the father of microscopy is. Option A is Antony von Leeuwenhoek. Option B is Jacques Jensen. Option C is Louis Pasteur, and option D is William Harvey. So you have a 2 minutes time. Okay, time is over. So let's see the answer for this question. So the right answer for this question is option A, that is Anton von Leeuwenhoek. So this he is the one like who discovered this microscope, Anton von Leeuwenhoek. So he also called as the father of microscopy. Why or also father of microscopy as well as father of microbiology. So this Anton von Leeuwenhoek, he was a Dutch scientist and also entrepreneur who invented this single lens microscope to study the microorganism. So basically, he was the one who first discovered and described the microorganism, which he called the animalcules, like animal-like. And other than that, he also discovered the RBC cells. um, how he make this microscope, basically he learned to grind the lenses and all, and then he, after the grinding of the lenses and all, he make this simple microscope. He crafted this lenses that allow him to see this single cell organism. And after that he made like 500 approx microscope for his like own research and all. Before that we can't even imagine and we can't even see the like single microorganism, simple microorganism. So after the discovery of this microscope by this Anton von Leeuwenhoek, he's totally changed the field of the science because because of his discovery of this microscope. Now we can able to see the simple like very small micros, a very small microorganism. So that time whatever he discovered the microscope, it was expected that it was a like with exceptional magnification for its time. And using his microscope he was the first to observe this described microorganism, and he recorded his observation in chair with the Royal Society of London. For these reasons and techniques, he earned this title that is called the father of microscopy. So let's move to the next question. The ribosomal RNA present in both prokaryotic and eukaryotic ribosomes. Option A is 16S rRNA. Option B is 18S rRNA. Option C is 5.8S rRNA. And option D is 5S rRNA. So which one is the right answer for this question? You have a 2 minutes time. So your time is over. Let's see the answer for this question. So the answer for this question is 5s rRNA. So let's see, like what are this ribosomal RNA? Basically, ribosomal RNA molecule in cells that form the protein synthesizes organelle. We called it as the ribosomes. So these ribosomes help in the protein synthesis, and where it is formed in the nucleus, and from there it is exported to the cytoplasm, that with so that to help the translate the information in messenger mRNA. And other than rRNA, there are two major type of RNAs, the mRNA and tRNA. mRNA is the messenger RNA and tRNA is the transfer RNA. So these molecules of rRNA are synthesized in the cell nucleus, and in the cell nucleus where it is formed in a specialized region of the cell nucleus, and we called it as nucleolus. And from there it appears as a dense area within the nucleus, which contains the gene that encode the rRNA. These rRNA basically have two different types. In prokaryotes it is of 70s type, and in eukaryotes it is of 80s type. So this prokaryotic ribosome is made up of two subunit, that is 30s, and the largest subunit is 50s. And eukaryotic ribosome is also made up of two subunit. One is small subunit, that is 40s, and another one is the large subunit, that is 60s. So this small subunit is further made up of this 16s rRNA, and the larger subunit of the prokaryotic ribosome is made up of 23s rRNA and the 5s rRNA, while eukaryotic ribosome is made up of small subunit is made up of 18s rRNA, and large subunit it is made up of 20 srRNA, 5.8 srRNA, and the 5 srRNA. So we can see that this 5s rRNA is present in both prokaryotic as well as eukaryotic. So the right answer for this question is the 5s rRNA. So, moving to the next question, the type of association where two organisms function as a single unit and help each other for survival is. So, the type of association where two organisms function as a single unit and help each other for survival is. So let's see the answer for this question. So option A is the correct answer. That is symbiosis, where the two organisms function as a single unit and help each other for the survival. That is, both are benefited with each other. So the symbiosis is a type of close and the long-term biological relationship between the two organisms of the different species. Here we can see the example of this clone fish. This clone fish feed on the small invertebrates and the fecal matter from the clownfish nutrients to the sea anemone. So these the fecal matter of this clownfish provides the nutrients to the sea anemones. And how sea anemone benefited by this clownfish? This clownfish is protected from the predator by the anemone stinging capsules. So this sea anemone is giving nutritions to this sea anemone, and sea anemone is preventing this clownfish from the predators because they have a stinging cells which secrete some type of chemical so that the predators can be away from this clownfish. So in this way, both are benefited with each other, and both organisms function as a single unit and help each other. And we also call it as a mutual kind of relationship, that is both are benefited for each other. So the right answer for this question is the symbiosis. So the next question is, the smallest unit in the taxonomic classification with fundamental similarities is. Option A is family, option B is phylum, option C is genus, and option D is species. So you have a 2 minutes time. Think about this question. Okay, time is over. Let's see the answer for this question. Yeah, species is the right answer for this question. So this is the taxonomic hierarchical classification. Here we can see that species, and then it will come two or more than two species come under the genus, then genus comes under the family, and then it goes to the higher, that is order, then into the class, and then phylum, and then kingdom, and then domain. So the smallest unit for any classification is the species. We can take it as an example of like mango. We called it as Magnifera indica. Here, indica is a species and Magnifera is a genus. So in this mango varieties, Magnifera is come like more than two like species are also there, which is under the like there are different types of mangoes varieties, and we can say that the different genus are there, but it all come under the same genus, that is Magnifera. So in this way, this species is the very smallest unit in the taxonomic classification with fundamental similarities. So the next question is, the protein rich layer in the cell body of Euglena that give flexibility to the organisms is. Option A is Hyaluronate layer, option B is pellicle, option C is collagen, and option D is S layer. So you have a 2 minutes time. Okay, let's see the answer for this question. Pellicle is the right answer. Pellicle is a protein-rich layer found beneath the plasma membrane of organism like Euglena. And here we can see that this is the Euglena, and this is the outermost covering of this Euglena. We called it as pellicle, and this pellicle is a protein-rich layer, and it is just present behind the plasma membrane. It provides the flexibility, and also it provides the structural support to the cells, and it's not that much rigid cell wall. Like plants have a rigid cell wall, right? But in this euglena, this pellicle material is very flexible, so that this euglena can twist and bend while maintaining its shape. So this protein-rich layer, we called it as pellicle, which is present in this euglena. This elrina layer is present in maize and all cereal crops. Collagen is present in the plants, and this S layer is present in the prokaryotes. So the right answer for this question is the pellicle. So let's move to the next question. The organism live in extreme salty areas is option A is Halobacterium, option B is Thermoplasma, option C is Pyrococcus, and option D is Sulfolobus. So you have a 2 minutes time. So let's see the answer for this question. So the organism which live in extreme salty areas is the Halobacterium. Halo, that is haline, that is salty areas. Thermoplasma, that is high temperature. Pyrococcus is, that is the low temperature. Sulfolobus, that is sulfur containing bacteria. So what is this halobacterium is? It is one of the most studied archean, that is archean is ancient bacteria, and it is salt loving because it found in this, basically it's found in the salt ponds at SC end of Francisco Bay, Dead Sea. It contains bacteriorhodopsin, that is a light sensitive pigment that gives halobacterium its color. And there is also a similar photosynthetic pigment similar to rhodopsin found in the vertebrate retina. So they have a similar kind of pigmentation which is present in the vertebrate retina. But this found to withstand lethal doses of UV radiation and extreme dryness and even the vacuum of the space. So it have a very special kind of characteristics, that is it is found to withstand lethal doses of UV radiation. If we'll, because we know that even in the hood, like when we are going for our work, before that we turn on the UV light so that it will sterilize all the surface area and wipe all the bacteria. But this halobacteria, even we give the high doses of UV radiation, still it will survive. It will it, it will not lead, this UV radiation will not lead to halobacteria death. So still it will survive. So that kind of, in the extreme environment also it will survive. And so the next question is the phylum that is closely related to both chordates and non-chordates and is considered as the connecting link between them is option A is Echinodermata, option B is Arthropoda, option C is Annelate, and option D is Hemichordata. So you have a 2 minute time. Okay, time is over. Let's see the answer for this question. So, hemichordata is the answer for this question. What is hemichordata? Hemichordates. That is half chordates. So, this is the half chordates means they have half characteristic features like chordates and half like the non-chordates. That's why it is the connecting link between chordates and non-chordates. So, what are those features which resemble with the chordates, and what are the features which resembles with the non-chordates? Let's see. So, the shared feature with chordates is the pharyngeal gill slits, and second one is the notochord-like structure. So, this hemichordates, like chordates, they have a pharyngeal gill slits, and these pharyngeal gill slits help the chordates to respire and help in the filter feeding. And the second one is the notochord-like structures. These hemichordate possess a structure called the stomochord. These stomochord is not a true notochord. That's why we called it as stomochord, which was once thought to be homologous to chordates, that is notochord. Earlier, it was believed that it's the same as a notochord, but then later it's found that it's not a true notochord. That's why it named it as stomochord, and it adds to the connection that it have a chordates like feature that is called the notochord, and we called it as a stomochord. And what are the features which resemble with the non-chordates? The non-chordate features, they have this body segmentation as like the non-chordates. These hemichordates share the body organization pattern such as simple nervous system and open circulatory system. But in chordates, all have this closed circulatory system. But in case of this hemichordates, they have an open circulatory system and which resembles to the non-chordates. Also, they have a similar larval form because in case of chordates, there is no larval form. They have a tornaria larva of hemichordata, and this tornaria larva is closely resembles to the bipinnaria larva of the echinoderms. So, which highlights is the evolutionary link with the non-chordates, as they have both features similar to the chordates as well as similar to the non-chordates. That's why they have highlighting the evolutionary link with the non-chordates as well as that will leads to the chordates. So that's why we placed it between two and name the phylum is the Hemichordata. So the next question is the radial symmetry is found in which of the following? Option A is Platyhelminthes, option B is Annelids, option C is Cnidaria, and option D is Porifera. Okay, so time is over. Let's see the answer for this question. So Cnidaria is the answer that they have a radial symmetry, but as we know that there are two kind of symmetry. One is the radial symmetry, and another one is the bilateral symmetry. So what is this radial symmetry? Is when we cut the body through the center, like wherever we will cut, like here from here, it will cut into the same pieces, like same segmentation, like both. It will give rise to the same like whenever we cut through this like around this central body axis, right? So that symmetry we called it as the radial symmetry. So that arrangement of similar part whenever we cut into the around the this central axis. And here we can take it as an example of this sea anemone, and another example is like starfish and all. Another type of symmetry is the bilateral symmetry. And so what is the bilateral symmetry is? This bilateral symmetry is when we body can be divided into the two identical half. Only when we cut it into these plane, then only in that plane only it will cut it into the two identical half. And example, we can take it as an echinoderm might have this bilateral kind of symmetry. So these are the two symmetry, right? So moving to the next question is, and here we can take this crab also. So next is, mollusks use their feeding organ called radula to scrape off algae from the rocks. The radula is made up of option A is lignin, option B is pectin, option C is chitin, and option D is suberin. So you have a 2 minutes time. So time is over. Let's see the answer for this question. So chitin is the right answer for this question. So the radula is made up of chitin, um, which mollusks use their feeding organ called the radula. Um, what about this first option, that is lignin? And lignin, we know that it is found in the plant cell wall, and in the plant cell wall, what it helps in providing the rigidity to the plants, so it's not present in the mollusk. So we can eliminate this option. And second one is the pectin. It is a plant polysaccharide which is found in the middle lamella of the cell wall, and it helps in maintaining the cell adhesion, and it is also not related to the mollusk. And chitin, we know that it is the right answer. And suberin is a waxy substance which is present in the plant tissue like cork, and which act as a water barrier. So the right answer for this question is the chitin. So moving to the next question, compound eyes are present in option A is octopus, option B is mosquito, option C is frog, and option D is human. So you have a 2 minutes time. So let's see the right answer for this question. So mosquito is the right answer. That compound eye are present in the mosquito. Here we can see that this is the mosquito, and it is made up of this very thousands of tiny units. We called it as omatida. So this compound eye is a visual organ found in basically mostly in arthropods, and as mosquito is also a part of arthropods, and also such as insects and crustacean and all. And it consists of like thousands of this omatida, and with these omatida captures a part of the visual field. So this omatida, let's say this capture this field, and this omitida capture this field. So these different omitida capture a different angle of the visual fields, so that it can provide the mosquito a wide angle view. And this wide angle view, so this eye, because of this wide angle view, this help them to detect the movement of threads from almost any direction, right? So the even they have a like single aperture eye. A compound eye have a poor image resolution, so they possess a very large view angle, and also this also rely on the detecting movements uh, to locate prey and avoid predators. So this compound eye allow them to spot even at the slightest moment. So they have a different kind of features. Like even the predators move like very slowly. Even then, they can detect the movement of this predator, and so that it can move away from that point. So basically, how the individual light receptor behind each lens turned on whenever like this ommetator gives a signal. Whenever this light reaches to the lens, this individual light receptor behind of each lens are then turned on and off, like whenever the intensity varies during the movement or during the objectivist movement, and then they created a flicker effect, and we know which we known as the flicker frequency also. So that which is the rate at which this omega are turned on or turned off, and which facilitate the faster reaction to movement. So in this way it works. So the next question is the common method of asexual reproduction in hydra is: option A is conjugation, option B is spore formation, option C is binary fusion, and option D is budding. So you have 2 minute timings. So let's see the answer for this question. So budding is the most common method of asexual reproduction in Hydra. So this is the Hydra. So here we can see that this is the parent Hydra, and then this bud is developing, and new bud will form, and then it will pinch off from the original and give rise to this offspring. So this is the most common method of asexual reproduction in case of Hydra, and we called it as it budding. Very simple question. So let's move to the next question. The type of organization found in the grandia, which is a kind of sponge that help in the flow of water through the organism is. So this grandia, which kind of this organization like water canal system is present. Option A is leuconoid type, option B is recon type, and option C is escon type, and option D is cycon type. So you have a 2 minutes timing. Okay, let's see the answer for this question. Cycon type. So the cycon type of organization is present in the grantia. So this is the cycon type of canal system, and it is also present in the cycon, which is another sponges, which is come under the second type of canal system only. Here, how this water moves, there is a entry which is the dermal ostia. The water enters through this dermal ostia, and then it will goes into the incurrent canals here, and through the incurrent canals this water absorption will be there and nutrient absorption will be there through this coinocyte cells, which is a special kind of cells. And through this then it will goes into this s- uh, middle part, and we call it as a spongiocele, and after the spongiocele it will goes out through the osculum. So in this way this water moves. This cyclone type body wall, as it is very thicker compared to the Ascon sponges. So basically there are three types of canal system: Ascon type, cyclone type, and Leucon type. So the very common very simpler one is the Ascon type, which is present in the asconoid sponges, and the middle one is the cyclone, in which the body wall of the sponges is very thick and it is folded into the radial canal. Here we can see that it is the thick walls and it is folded into this radial canals, and that open into the central cavity, and we can see that this radial canal open into this central cavity we called it as spongiocele. These radial canal are lined with the coinocytes or collar cells, and these radial wall are lined with this this tiny yellow like structure. We called it as coinocytes. So these radial canals are lined with this coinocytes, that is a special kind of cells which is present and it help in the water circulation. So this coinocytes or we called it as collar cells, help in the water circulation as well in the food capture. So what are the opening present in the second type of canal system? First is the ostia, that is the small pores on the surface and through which the water enters. We call it as dermal ostia, and it will leads to the incurrent canals. These incurrent canal pass water into the prospiles, that is a small opening that connect to the radial canals. And after that, apopiles opening at the inner end of radial canal, we call that those opening apopiles, through which water passes into the spongiocells, and through the apopiles this water goes to the central area, we call it as spongiocells, and through the spongiocell it goes out of the body surface through a big opening, small large opening, we call it as osculum. How this water flows in the second type of canal system: water enters through the ostia, then it will goes to the incurrent canal, and then prospiles, that is a small opening that connect to the radial canal. So through this opening it will goes to the radial canal, and in the radial canal they have inner opening, we call it as apopiles, and through the apopiles it will goes to the spongiocells, and after the spongiocell it will goes out through a big opening or a small large opening, we call it as osculum. So in this way, this water flows in the second type of canal system. So as I already told that what is this coinocytes, that are special kind of cells which help in the water circulation and as well as the food capture. So these line the radial canal and it create the water currents by beating their flagella. They also trap and engulf the food particles through the phagocytosis. So other canal system present in the sponges is the Escon type of canal system and the Leucon type of canal system. So this Escon type canal system is the simplest type and it is seen in the small sponges. So water flows directly into the spongiocele, that is the central cavity, through the ostia and exit through the osculum. So they were very simple kind of canal system that water enters and come into the spongiocells and goes out through the osculums, and these coinocytes line the spongiocells. So this is the very simplest type of canal system, and other is the Leucon type. It is a very complex canal system and it is found in the large complex, and here multiple chambers are present, that we call those chambers are the flagellated chambers, where these coinocytes are located. So water flows through the canal into this chamber so that it helps in the more efficient filtration before it exits through the osculum. So this is the Leucon type of canal system. So let's move to the next question. The coiled tubular duct-like organ which filter and remove the waste from earthworm is: option A is conadial cavity, option B is pericardial cavity, option C is nephridia, and option D is protonephridia. So you have a 2 minutes time. So let's see the answer for this question. Nephridia is answered that these nephridia are the excretory organs which is basically found in this annelids, or we call it as earthworm, and they consist of a tube with a nephrostome opening and nephritiophore, that is the exit to filter the waste from the coelomic fluids. So here one can puzzle like in the nephridia and protonephridia, but these protonephridia are the simpler excretory organ, and it is basically found in the flatworms. And this nephridia is present in the earthworm. So this protonephridia have a tubules with the flame cells, and they use the cilia to filter the waste and excess water. So the tubular organ which filter and remove the waste from earthworm is the nephridia, not protonephridia, because protonephridia is present in the flatworms, and we also called it as a flame cells also. So this is the like nephridial system of this earthworm. It consists of a tubular system that opens internally into the coelom and through a funnel-like structure called the nephrostome, and externally by an opening called the nephridiophore. So, so moving to the next question, a flowerless plant with no true roots but rhizoids, that is root-like structure, and produce the spores in a small capsules. Option A is a grass, option B is mosses, option C is ferns, and option D is maize. So you have a 2 minute time. So let's see the answer for this question. This is a simple one. So mosses. So this is the mosses. Here we can see that it is a non-flowering plants, and it lack the true roots. So they don't have the true roots. They have a root-like structures. That's why we can easily remove it off, right? The grasses, when we try to like when we apply the force, we need very small force to like detach this mosses from the substrate. Because they don't have a true roots, they have a root like structure, we call it as rhizoids, which are the hair like structures, and that help anchor the plant to the substrate, and through this rhizoid only it absorbs the water and nutrients. This mosses reproduce by producing the spores, which are contained in a small capsule-like structures at the end of a stalk-like structure, and we call this body is called as a sporophytes. Basically, this mosses reproduce by producing the spores, and it is formed through this sporophyte structures. Unlike the flowering plant, mosses don't produce any seeds, but what they do is they release the spores which will germinate and grow into the new plants. So the right answer for this question is the mosses. Moving to the next question, an egg-laying mammal with a beak-like bird is option A is echidna, option B is duck, option C is kangaroo, and option D is king cobra. Very simple question. Okay, let's see the answer for this question. So, echidna, it is a type of egg-laying mammal, and we also called it as monotrim. It is a spiny burrow animal which is found in the Australia and New Guinea. These monotremes are the unique because unlike the most mammal, as we know that the mammals give rise to birth, the young ones, right? But these echidna, they lay the eggs instead of giving birth to live young. After laying the egg, the mother incubates them until they hatch. So this is a exceptional to the mammal, which is an egg-laying mammal with the beak like a bird. And here we like how it look like. It look like this. So this is the echidna, and here we can see that they have a spine-like structure. Spiny structure is there, right? And also they have this beak like a bird. They have a beak-like structure. This is the egg-laying mammal, and we call this is echidna. So moving to the next question, a correct characteristic feature of a heart and blood circulation present in mammalia is option A is incomplete three chamber and double circulation. Option B is complete two chamber and single circulation. Option C is incomplete and chambered and double circulation. And option D is complete four chamber and double circulation. So what is the right answer? That is present in the mammalia. Okay, let's see the answer for this question. That is complete four chamber and double circulation. So mammals have a complete four chamber. That is, they have a four chamber: right atrium, right ventricle, left atrium, left ventricles. So they have this three. They have this all the four chambered, and double circulation will be there because they have a different oxygenation and oxygenation process. So it will goes into the double circulation process. And here this right atria connect to the right ventricle through this tricuspid valve, and left atria connected to the left ventricle through this bicuspid valve. So this human, that is mammalia, have this four chamber and the double circulation type of bl- so then moving to the next question, the type of scale present on the Bombay duckfishes. Option A is placoid and cycloid scales. Option B is placoid and tenoid scale. Option C is cycloid and tenoid scales. And option D is only placoid scales. So you have a 2 minutes timing. Okay, let's see the answer for this question. That is cycloid and tenoid scales. So this Bombay duckfish have a two kinds of scale. That is, one is the cycloid and another one is the tenoids. Like they have this two types of scales. The cycloid scales, they give a smooth, round, and thin scales with a uniform surface. These scales overlap and give a sleek streamlined appearance to this fish, and they are mostly present in the salmonids, that is salmon and trout. They have a more flexible and smooth. Antinoids like scales, how it is similar to the cycloid, but they have this spiny or comb-like structures, and these spines give them a rough texture. Here the cycloids give a streamlined appearance, and this stenoid scales gives a rough texture. And typically, the stenoid scale present in the Persian and they have this comb-like projection, we call it in eye, which help in reducing drag and protecting the fish. But this Bombay duckfish, they have a both type of scales, cycloid as well as this tenoid scale, smooth as well as this rough surface. So moving to the next question, the two pair of limb in the frog arises from option A is pectoral and ventral region, option B is pelvic and dorsal region, option C is only pectoral region, and option D is pectoral and pelvic region. So what is the answer for this question? So let's see the answer for this question. Pectoral and pelvic region. So the pectoral, that is as we know that the forelimb arises from the pectoral and the hind limb arises from the pelvic region. So this is the frog structure. This pectoral, it will give rise to the forelimbs, and this hind limb is through this pelvic region. So the two pair of limb in the frog arises from pectoral and pelvic region. So moving to the next question, which is the field of science that deal with the human behavior in contrast to mental processes? Option A is history, option B is linguistic, option C is psychology, and option D is public administration. So you have a 2 minutes time. So let's see the answer for this question. So psychology is the right answer for this question. As psychology is the scientific study of human behavior and mental processes. While it focuses on understanding the thoughts, emotions, and behavior, it also distinguishes between the observable actions, that is behavior, and internal mental processes. So the psychology is the field of science that is deal with the human behavior. So moving to the next question, the book that explains how species evolved over time and gradually diverged from each other through a process called natural selection. Option A is origin of life, option B is origin of species, option C is origin of virtue, and option D is selfish gene. So you have 2 minutes time. So let's see the answer for this question. That is origin of species. So this is by the Charles Darwin. So he wrote this Origin of Species, and he described that how the species evolved over time and gradually diverged from each other, and through a process called the natural selection. So the right answer for this question is Origin of Species. So that's all for today. Thank you.